Hello everybody, my name is Flavio Pazos from Uruguay and I'm going to present you a work entitled Gene Function Prediction in 5 Model Eukaryotes, exclusively based on gene relative location. For allowing me to show you our work. First of all, I would like to thank the organizer that was published last year. Let me start giving some background of most genes. In fact, less than 1% of known sequences have any experimental validated function. The figure shows here in red the number of sequences in UniProt and in blue the number of sequences with at least one experimentally supported functional annotation. Note here the logarithmic scale in the y-axis. We can see that the proportion of genes with experimentally validated function is very small, and that proportion will continue to decline in the future, so improving automatic functional annotation is crucial. The gene ontology is a representation of all possible functions of the products of gene expression. It is organized in 3 graphs, 3 directed acyclic graphs, the biological process graph ontology, the molecular function ontology, and the cellular component ontology. These 3 graphs represent 3 aspects of the biology of gene products, and each of them is made up of Go terms and their hierarchical relationships. Here, we represent the genes as consecutive squares along the genome and the functional annotations as colored labels that can be assigned to genes. Automatic functional prediction use the available data to establish putative association between genes and gene ontology terms. Over the last 2 decades, several methods for functional prediction have been developed. These methods are evaluated and compared in the CAFA challenges, that are competitions in which the participating teams formulate functional predictions, and after a pre-established period of time, these predictions are evaluated using the new experimental evidence and compared to 2 baseline methods, BLAST and naive-Bayes. In these competitions, the best results are obtained with machine learning-based methods and combining different types of predictive variables. Machine learning models can recognize shared patterns between genes with the same function. These models are able to combine different predictive variables, typically including sequence and sequence-derived features, and use different algorithms and architectures to predict association between genes and Go terms. These models produce catalogs of candidate genes that can guide traditional experimentation, saving time and money. So, which is the motivation of this work? Was to look for new ways to extract relevant information from the available data in order to improve functional prediction and to explore an interesting biological hypothesis that gene relative location can predict gene function in eukaryotes. We know that prokaryotic genomes have operons, and gene location of prokaryotic genes has been used to predict their function for more than 3 decades. On the other hand, until the beginning of this century, it was assumed that eukaryotic genes had a random distribution along genomes. But plenty of evidence began to accumulate during the last years indicating that eukaryotic genomes are not randomly distributed, and pointing to the existence of conserved distribution patterns for functional groups of genes. So we asked ourselves, could we use these distribution patterns to infer gene function? How did we address this question? We started by defining the local enrichment analysis, consisting in calculating the overrepresentation of a Go term in the group of genes surrounding a gene of interest. For example, if we focus on this gene and define a window of a given size W, we can calculate the enrichment in red genes that is found in this window. We calculated the functional enrichment in different Go terms around each gene, considering different window size. Then we constructed these arrays that we term the functional landscape arrays, or FLAS, that contain in each row a different window size, and in each column a different Go terms. And in each cell we put the enrichment found for that Go term in that window in the surroundings of that genes. Walking along the chromosome, one gene at a time, we calculated the functional landscape array of each gene. Our hypothesis now is that the genes with similar biological functions have similar functional landscape arrays. To test this hypothesis, we used these arrays to train a set of multi-class hierarchical classifiers and predicted functions in 5 model eukaryotes: the yeast, the worm, the fly, the mouse, and the human. We choose these organisms because they are well annotated, they are diverse in terms of genome organization and phylogeny. We implemented one model for each pair formed by one of the 3 ontologies and one of the 5 organisms, and used the functional landscape arrays as the only predictive feature. Here I show some details of the implementation of the models. For each pair formed by an ontology and an organism, we train a binary classifier for each Go term associated with at least 50 genes to be able to use at least 40 genes to train and 10 genes to test. The negative examples of the training sets were selected using the siblings policy, and we used the node interaction method to fulfill the true path rule. To train our models, we used the experimentally validated annotations available on November 18, and to evaluate the models, we used the new experimental annotations accumulated up to September 21. To evaluate the performance of the models, we used the hierarchical F1 score. Let's see now some results. In each graph here, you can see the hierarchical F1 score as a function of the classification threshold. The continuous line corresponds to the hierarchical F1 score of our models, and the dashed lines to the models that assign the Go term frequency as a prediction score. This is one of the kappa baseline methods. In each column, we have one organism. In each row, we have one ontology, biological process, cellular component, and molecular function. We can see that in all cases, the performance of the model exclusively trained with the functional landscape arrays is higher, and that in each case, the difference reaches a maximum at a different classification threshold. We can also see interesting differences between ontologies and organisms, which we can discuss later. Here we compare the performance of our models with that of BLAST, the other baseline method used in the kappa challenges. We were able to compare our results only for the fly, the mouse, and the human, since the available data only includes results for these organisms. Surprisingly, in the 3 organisms, our models outperform BLAST in the biological process ontology. Something you can see here, here, and here. And also, in 2 of the organisms, our model outperformed BLAST in the cellular component ontology. Here and here. Here we see for each ontology the depth in the graph of the predictions we obtained. For biological process, for cellular component, and for molecular function, each colored line corresponds to one of the organisms, and we can see that biological processes not only seems to be more predictable from gene relative location, but also that the predictions are deeper and thus more specific. The main conclusions of this work are that we found that gene relative location can be more informative than sequence to infer gene function, particularly when predicting biological process growth terms. That we found levels of organization thought to be exclusive of prokaryotic genome and its characteristic operands, and that we obtained thousands of new functional predictions, some of which have been already experimentally validated by others. Finally, I would like to thank all the team members, Diego, Pablo, Patricio, Gustavo, and Rafael, to thank all the funders, and to thank you for listening.